I'd like you to imagine the following scenario. Suppose you're driving to work one morning, you've come to a stop at a stoplight, and all of a sudden sirens start sounding all around you, and a police car pulls up on your right, and another car pulls up in front of you. The policemen jump out and they say, "Sir, we'd like you to get out of your car. Are you such and such an individual?" You answer that you are. They immediately put handcuffs on you. They drag you off to jail. Now, what you find out is that you have been accused of murder, and you are the prime suspect. The police supposedly find all sorts of evidence that implicates you, and the day of your trial arrives. Now, you've hired the most expensive lawyer you know, highly competent. You have an airtight alibi, so you're not even worried. The prosecuting attorney gets up first, and he just presents the evidence after evidence after evidence for your guilt. The jury listens in rapt attention, and they start to increasingly look at you with anger and judgment in their eyes. Well, finally, the prosecuting attorney ends his case, and it's time for the defense lawyer to defend you and lay out the defense before the jury. Your defense lawyer stands up prepared to make your defense when the judge says, "Stop right there," bangs his gavel on the bench and says, "I think we've heard enough. We've heard plenty of evidence for the jury to come to a decision. Now I'd like to instruct the jury to go and deliberate on what they've heard and come back with a verdict." What would you think of that judge? I think, if you're honest with yourself, it's the judge that should be thrown in jail, not the defendant, because only one side was allowed to be heard. Do you understand what has been going on for over 2 generations throughout America? In every school, in every university, the students are only allowed to hear one possible viewpoint. You see, there are only 2 possibilities for the outcome of a murder trial. Either the defendant did it and is found guilty, or there's not enough evidence, or in reality he did not do it and he's going to be found innocent. Even a hung trial can be retried to find guilty or innocent verdict. In the case of where did everything come from? Where did the written language of DNA come from? Where did life come from? Where did the time, matter, and energy of the universe come from? There are only 2 possibilities. Either it all made itself, the DNA code wrote itself, bacteria turned into human beings, time, matter, energy, it just appeared and made itself, or there is a creator, there is a God, there is something outside of the time, matter, space universe that organized it, that created it. Students are only shown the evidence that leads them to the belief that everything made itself. Cosmic evolution, chemical evolution, biological evolution. They are never allowed to see or consider the problems with this idea or the evidence that supports the reality that we have a creator. It's exactly like what you just considered. The judge is the American legal education system, and it only allows one viewpoint to be seen by the students. So obviously, generation after generation, those students grow to become the teachers, they become the professors, they write the textbooks. This idea gets locked stronger and stronger into the minds of the people only allowed to hear that one viewpoint. This is the travesty of what's going on all around us, and it's why the idea of creation is considered so ridiculous to so many people. Not that it's not true, the evidence for it just doesn't get to be seen. We're here at the Midland Center for the Arts, where there's a traveling Lego display to talk about the complexity of the individual parts which the human body and all biological life is made out of. Now, where did the parts of life come from? There's an incredible analogy to that. Lego blocks. I brought a pirate ship. As you look at the ship, you'll notice it's made of lots of tiny little blocks, different sizes, different shapes, different colors, but it also has some specialized parts, cannons and sails and so on. And I think about every young child has played with Legos from one time or another, and you'll notice how they connect together. You have to follow a specific set of instructions before you can form the final structure and put all the individual parts together to form the pirate ship. Now, the same is true of life. The most common building block, which our body and every other living organism is made out of, are small molecules called amino acids. We're going to talk in a little bit about those amino acids and how they link up to form proteins that form, in essence, the Lego blocks of our body. In this case, we have a completely different Lego structure. Here is a 747, in essence, a different mode of transportation, much more advanced. And yet, the individual structure is made out of many of those exact same Lego blocks, the same sizes, the same shapes. They've just been fit together to form a different structure. But there are also, again, some specialized parts. The wings are not found on the pirate ship. The jet engines are not found on the pirate ship. Those are totally new structures that just appeared to make the 747. Okay, now we have an even more advanced mode of transportation. We go to one of the Star Wars films where we have the Slave One spaceship. Now you'll notice this one has some sort of super advanced engines. It flies through the air at incredible speeds, and yet most of this structure is made out of those same individual little building blocks that are lined up and reoriented in a different way, and again with some specialized parts. So how did these structures arise? How did you go from the pirate ship to the 747 to the Star Wars spaceship? Well, if evolution happened, then somehow random parts just modified themselves to make more specified parts, and somehow they just fell into place to form these different structures. You understand that the body structures of different types of organisms are very, very different and much more complex than these Lego models. These are pure simplicity compared to the incredible complex molecules of life. But there's an even bigger little secret of evolutionary biology. Now, students in high school biology class, they don't ever even hear about this. It shows up as they get into the upper level classes in college. But there is something, as geneticists have studied life, called orphan genes. Well, there are genes that seem to just appear in certain body structures of certain kind of animals. Now, I attended a lecture by Dr. Eugenie Scott a few years ago where she stood up in front of a large college audience and said, the foundational principle of biology, which all biologists believe, is the common descent of life. Well, that's evolution. That means it took the coded information on DNA and it was slowly modified, and the individual parts were slowly transformed into another part. Now, the parts of our body, the most common one is called a protein, and the information to make the protein is coded into a specific gene. The DNA code is like a library. Appear. They're just there. Now, you would think if the pirate ship slowly transformed itself into the 747, there'd be a slow modification of specialized parts. You see, the creator of Lego, he took a common raw material, the individual blocks, to make the structure. But then to make specific structures, he created new parts, and they were just instantly there. The exact same thing is true in life. That's what orphan genes are. Because we have a creator, because they were created, because God made them, That's why they exist. So where did the individual parts of our bodies, of every biological creature, come from? What are called the amino acids or the proteins that make up our body. In every biology book in the world, any student who has ever taken biology, they will hear about something called Miller's experiment early on in that biology book. Now, Miller's experiment was done in 1952, and he created some of the amino acids that are found in life. Now, these amino acids are kind of like Lego building blocks. They come in different colors. These particular ones, they all have the same shape because amino acids are quite similar, and they will link up in a certain order. Yellow followed by 2 reds, followed by a blue, followed by a green, and so on. And when he did this experiment and added the energy, poof, the chemicals combined to form the various amino acids, which by analogy we're calling the Lego blocks. Now, you'll notice the Lego blocks are made to link together, and chemically, amino acids will also very easily link together. And when they link together to form a chain, it's called a protein. Now, it's the specific order in which these amino acids link up that determine how it is going to form the various parts of our body. Now that sounds real good. Wow, an explosion created the amino acids. The amino acids can link up to form proteins. Our bodies are made mostly of proteins. We've discovered how life formed, and that's the impression students are left. But again, there's this little secret that isn't even mentioned to the students. There are enormous problems with this experiment, and they're just glossed over or not even mentioned. Let me just mention a few of them. First of all, you'll note I said he left oxygen out because you don't get the Lego blocks if there's oxygen present. And yet as far down as we go into the rock layers of Earth, even down to where there supposedly wasn't any life on Earth, you find oxidized minerals, oxidized iron, oxidized rock. There was always oxygen here on this planet, and that's what the evidence shows, but it was just left out. Second, you'll notice the Lego blocks come in lots of different colors. There are 20 different amino acids that make up the proteins that make up our body. And yet he never got all 20 colors. He only got about 6 of them. Only 6 of the different colors of amino acids. They've never been able to show where did the rest of those amino acids come from. And you can't get life without them. The biggest problem, which is just totally left out, along with those amino acids, along with the Lego blocks, he formed formaldehydes and all sorts of other chemicals which actually will react with those amino acids and stop them from linking up. It's like blocks with a rounded edge. You see, when you put one of these formaldehydes or other chemicals on top of the amino acid, you can no longer continue to link up other ones. And there were far more of these chain stopping molecules than there were amino acids formed. It is literally, absolutely impossible for life to have formed in this way, for the proteins of life to have formed in this way, because you've got the wrong kind of amino acids. You got right-handed and left-handed amino acids. Life uses only left-handed amino acids, L-amino acids. It's like having a drawer full of gloves, random right-handed, left-handed gloves, millions of gloves, and then you reach in to grab a glove, and every time you reach in and you've got 50% left-handed, 50% right-handed, you reach in blindfolded to grab one, every time, millions and trillions of times, you only get the left-handed glove. It could never happen. You understand what Miller's experiment actually shows? Life could not possibly have formed in this way. That experiment was almost 70 years ago. For almost 70 years, the same evidence is shown to student after student, generation after generation, textbook after textbook, and the problems are left out, and it's implied this shows how life could have formed all by itself. What that experiment actually shows is life could not possibly have formed in this way. And it's the best that evolutionary biologists have ever been able to come up with to try to explain how life could have been made without God, when it actually shows life could never have formed in this way. You see, we've got to be showing these kind of things to the next generation, or they're going to be sucked into this idea that life just made itself. So we have talked about orphan genes, the idea that there are parts of very different creatures that just appear with no ancestral relationship. We've talked about amino acids and the things that are left out of the textbooks that show even the classic Miller's experiment from 60 years ago does not explain where the amino acids came from. Now I want to take a moment and talk about the molecules of life, the very parts with which our bodies are built. You see, the amino acids that I talked about are like different colored little balls. Would be one amino acid orange, another amino acid might be blue, another amino acid might be yellow. There's 20 different colors. And we don't even know how to make all of them. We don't see them occurring naturally. But let's assume there's only 5 colors, just for the sake of illustrations. So now you're trying to link them together to make what's called a protein chain. Our bodies are made out of hundreds of thousands of different arrangements of the balls. The balls link up to form chains. The chains are called proteins. But they have to be in a very specific order. If the colors are lined up in a certain way, it might spiral to make like a hair molecule. If they're lined up in another way, they might make the fluid inside of our eyeball. Another way might be the skin or the texture of the skin of part of a skin cell. Even a skin cell would have thousands of different proteins that make up its parts. So the question is, could these amino acids that are just somehow floating around out there, even though we don't find the right kind, we don't find all of them we need, somehow link up to form the correct proteins that could then go on to form parts of a bacteria which eventually become a person? Could chance processes make this happen? Well, we could do an experiment to figure out if this is true or not. Now, for simplicity, let's pretend this is an amino acid. It's just got 5 different proteins, 5 different colored amino acids. The typical protein chain in our body has several hundred specific amino acids in exactly the right spot. But could random chance produce a protein chain with just 5? Well, let's say here's our random arrangement of amino acids. They're all just floating around. If it's going to be an evolutionary process, it has to be blind. There's nothing guiding it. Now I am blind chance. Let's see if blind chance can produce a useful protein that has the right amino acids in the right spot. So we'll put the first one, which I grabbed, in the very first spot. Okay. Had to be in the right spot or it's not useful. And then we'll put the second one in the second spot. Reach way down deep, we'll grab a third one, and it just links up there naturally in the third spot, the fourth one here, and then the last one we'll place in the last spot. So I have now created a protein. Let's see if it has a useful biological function, because if the amino acids aren't in the right spot, it's a detriment. It will die. It's of no biological purpose. So what do we have? We needed a blue one first, it was yellow. A red one needed to be second, it's yellow. A yellow one needed to be third, it's red. Then we needed purple, and then we needed green, and it's red. None of them are right. Not a single amino acid ended up in the right spot. Now, statistics is science. You can determine using mathematical scientific statistics, is this possible or not, to produce even one molecule needed for life. You see, only once out of 20 times will you get the right color in the right spot. And it is once in 20 times once in 20 to get 2 in the right spot. Actually, to get just 5 molecules lined up correctly would only happen once every 3 million tries. Now, if the sequence was 20 amino acids long, it would only happen once every 100 trillion tries. That's 1 with 24 zeros after it. Only one time would you be likely to get 1 of those molecules right. The average protein in any organism is about 200 long. That would be more tries than every single atom in every molecule in the entire universe. It is absurdly impossible to even get 1 amino acid that's biologically useful by random chances. They are designed by an unbelievably intelligent designer. It's the only realistic, logical, consistent scientific explanation for how the components and parts of life have come into existence. We're going to take a moment now and talk about the complexity of DNA itself. We've talked about the protein molecules and how they have to be lined up in such a perfect sequence to be of any use at all to a biological organism. But what about DNA? How could DNA have made itself? DNA turns out to be the most compact information storage system in the universe. Unbelievable amounts of data and information stored in the sequence letters of information of that molecule. Now, DNA is made up of 4 chemicals that are lined up in a given sequence. In that sequence, through a very complex set of intermolecular biological systems and little information transferring mechanisms, is used to make the proteins, to line up those amino acids, to make the proteins, which then make the parts of our body. But how could that information have gotten onto DNA? Even when DNA was first discovered, it was acknowledged as being literally a miraculous molecule and system for containing, carrying, and transferring information. But from what we thought it was like to begin with, it's like a kindergartner's first reading book compared to what we now know about it. It turns out that the information sequence on the DNA molecule is not like a book where you would just read across each line and read to the next line and read to the next line and gather information. It turns out we have now discovered that the letters in DNA that are used to make the proteins of life can actually be turned on and turned off. You can start in the middle of a sentence and make a certain sentence to mean a certain thing, and pick up part of the next sentence and have it mean a completely different thing. You can actually read the letters forward and backwards to make different proteins in different sequences. You can turn those letters on and off by something that seems to be outside of the DNA molecule itself. It's called exogenetics. We are finding level after level after level of information containing systems inside of that molecule stored in a 3-dimensional structure where how the DNA winds itself determines how the letters are connected in order to make the right protein molecules. Now, I went to a conference at Cornell University dealing with information and the information content of DNA. And to be honest, I'm not a geneticist, I'm not a molecular biologist, and most of this stuff was like 3 stories above my head with all the technical language. But one of the speakers was really good at bringing things down to an analogy level to where it could be understandable. And he used this analogy for our current understanding of DNA. Now, suppose I have a book and I'm reading through it. It turns out to be an adventure novel. And man, I'm just reading page after page. I can't wait to hear how the hero gets out of one jam and into the next and how it all ends up. And man, it was just a great book. So I passed it on to a colleague at work who happened to have been from Germany. And he takes it home and comes back the next day and he says, "I stayed up all night reading that book. It was phenomenal. It was such a great technical book on the building of factories." And I'm thinking, the building of factories, it was an adventure. Well, let me see the book, see if you read the right one. He said, "Oh, it was such a good book. I gave it to a friend. He happens to be from France. He works down the hallway." So the next day, I went to his office, asked him if I could have my book back. He said, "Oh, the book was such a wonderful. It was a tragic romance. We love tragic romances in France. And it was written in French." I said, "Written in French?" It was written in English. And the German said it was written in German. He said, "Yes, I passed it along to my friend in Spain." So I tracked him down. He said it was a comedy written in Spanish. Written in English, in German, in French, in Spanish. You see, they all use the same English letters, but they use the letters to line up to create different meanings on the same page. Now imagine an author who could use the same letters laid out on a page
read through, and it would have one meaning in English, a different meaning in German, a different meaning in French, and a different meaning in Spanish, and make perfect sense, the same sequence of letters in the book on every page. It would be an intellect of incomprehensible ability that could write such a book. That is what the DNA molecule is capable of. You get different proteins from the same area of the same molecule, depending on whether the information is pulled out forwards, backwards, at the beginning, in the middle, at the end, to create different molecules from the same information in the DNA. Unbelievable. Now imagine natural selection and mutations just randomly changing the letters to create some new information. It would have to change it in such a way that it still made perfect sense in all 4 languages simultaneously. It's an impossibility. It could never happen. Now let me give one more example of the complexity of DNA. If you look at DNA, I want you to think of it as a zipper. You know, this zipper happens to have 4 different colors, like DNA has 4 different molecules. In this case, I'm using green, yellow, red, and blue to represent the 4 different complex chemicals that make up DNA. And they're lined up in a certain sequence pattern. Like Morse code is a dot, a dash, and a space. But with just a dot, a dash, and a space, you can create the entire language, knowledge base of humanity. So languages don't need 26 letters. They just have to have symbols that are non-repeated and have meanings assigned to them in a certain sequence order. Every definition of a language fits the DNA code. Well, it turns out, as I said, in order to create the proteins that make our bodies and make many of the parts of biological organisms, you literally have to unzip the DNA. Now you have open access to the information. Another molecule comes along and attaches itself to the DNA, extracts the information, makes a copy of it, like a negative version of this. It goes and then grabs amino acids and makes an exact sequence pattern that turns into a protein, which is used by the cells of our body. But in order to get the sequence off of DNA, you have to unzip it. The only way to unzip a zipper is with a zipper pull. Otherwise it won't come open. I mean, you can tug on it all you want. You can bang on it. You can pull it and snap it. It doesn't come open unless you have a zipper pull. Turns out this zipper pull is a protein. Now, I don't know if you know where I'm going with this, but I want you to think about it for a second. In order to make any protein in our body, you have to unzip the DNA in order to get the information to make the protein. But in order to unzip DNA, you already have to have the perfect sequence of amino acids to form the protein to unzip it. So you can't unzip it without a protein, but you can't make the protein without unzipping it. And you can't unzip it without the protein, and on and on and on, round and round. You can't do one without the other. They have to all be there at once. It's like the old riddle, which came first, the chicken or the egg? You understand, you can never get a chicken without an egg, and yet every chicken that is born and comes out of an egg already has in its ovary the eggs it needs to make the next chicken. You see, inside of the chicken is the egg. It's already there, and you can't get the chicken until you have the egg. So which came first, the chicken or the egg? It's a riddle that only has an answer if you realize they were all created simultaneously. That leads into the next subject, where we talk about irreducible complexity, how unless all the parts are there and they're all there at once, no form of life could exist. But before we go there, I want you to see this little film that shows how complex the inner workings of a cell really are, because it just drives home the reality of the awesomeness, the brilliance, the creativity and inventiveness of our Creator and our Savior who made us all.
Throughout this sequence, we've shown the incredible complexity of life, how the very parts of life had to have been created. They couldn't have just made themselves. You know, that's part of what God has told us in Romans chapter 1, starting with verse 18. He goes on to say, mankind is totally without excuse for belief in Him because of what He has made. When we see it couldn't have made Himself, we know God must have made it. It's the only other option, because God wants us to know the truth. But I think there's an even more powerful example from looking at the biological world of how we can know things couldn't have just made themselves. It's a concept called irreducible complexity. It was first coined by a professor by the name of Dr. Michael Behe. He wrote a book called Darwin's Black Box. It's probably coming up on 20 years ago now, but the concept is as accurate and powerful today as it was then. He used the example of a mousetrap. A mousetrap has multiple parts. Actually, there's not hundreds of parts like the simplest form of life. It only has 5. It has a platform. It has a hammer, which would come over and trap a mouse. It has a catch that holds the hammer back, a spring that springs it forward, and then a small little place to put the bait, a trigger. 5 parts. If any of those parts aren't there, it's worthless. It's a piece of junk. It doesn't do anything. They all have to be there. In episode number 1, we looked at a feature of a small insect called the leafhopper. Inside that insect, there's a couple of gears that make sure the legs move in perfect unison. If those gears aren't totally functional, completely designed to interlock with each other, attached to the legs with nerves and ligaments, everything needed to cause them to move, all the parts have to be there, it wouldn't work. You know, metamorphosis of a butterfly or a moth. Think of some sort of a pre-caterpillar worm that somehow developed the ability to spin a cocoon and then lock itself inside of the cocoon. It's just going to die. There's not going to be no next generation of butterflies. It has to have everything there to totally dissolve its body, completely rearrange its molecular structure, and come out with things that didn't even exist before it went into the cocoon. Wings and different sorts of legs and all the instincts and mechanisms needed. They all have to be there. 2 more examples, because God has made this so obvious. And this irreducible complexity where all the parts are needed is seen everywhere throughout the biological world. But let's look at another example. You know, I have a fan, and it's kind of heavy. And, you know, I hate carrying the thing around. So, you know, I think what I'm going to do is just kind of like get rid of some parts. I'm going to, you know, get rid of the cover. Well, that'll save some weight. And, you know, it still spins just fine. Take the back off. And I don't know, it's got all this stuff in here. It's like, what do you need with all these extra wires? You know, I'm going to get rid of this thing right here. It just really just kind of hanging around. Doesn't seem to be serving any purpose. There, got that off of there. That saves some weight. And, you know, this gear, it's like there's 2 of them. One ought to be enough. Get rid of that baby. Okay. There, it's quite a bit lighter now. Oh, it's definitely lighter now. All right, so I got a lighter fan. Didn't need all those extra parts. Now I'll just plug her in and we'll just see how well it works. Huh. It doesn't work unless all the parts are there. Now you think, yeah, but obviously a fan, you know, it's got an electric motor, it's got magnets, you know, it creates positive, negative charges to get it to spin. You know, it's obvious it has a designer, but, you know, life, the parts could just come together. Well, there's an exact analogy in the biological world. There are electric motors 1,000,000 times smaller than this. You need an electron microscope to see them. They're inside of a bacteria. They have all of the parts of an electric motor on a molecular scale. You know, the rotor and the stator, the magnetism, the shaft, the little thing that flips around is called a flagellum, and it spins at 10,000 rotations per minute, can stop in a quarter rotation in reverse and go back the other way. Mankind has never built anything with those kind of capabilities. And if all the parts aren't there, it can't work. Scientists have studied this extensively. They've identified 40 different proteins. Now remember the proteins? They're a long chain of specific amino acids. Those amino acids line up in one sequence to form this flagellum, which is like the blade, another sequence to form the positive stator and the rotor, the shaft, but all 40 have to be there, or just like my fan, you remove one part, it doesn't work. Now, what do evolutionists say about this? How did all those parts get there? How did they get in the right place? Where did the proteins come from? They say, well, they just co-opted them from another organism that was using these proteins for another purpose. I want you to watch this little video that shows how ridiculous that concept is and how obvious God has made it, that He is the designer behind all of life.
Scott Minnick has studied the flagellar motor for nearly 20 years. His research has led him to challenge the co-option argument.
With a bacterial flagellum, you're talking about a machine that's got 40 structural parts. Yes, we find 10 of them are involved in another molecular machine, but the other 30 are unique. So where are you going to borrow them from? Eventually, you're going to have to account for the function of every single part as originally having some other purpose. So you can only follow that argument so far until you run into the problem of you're borrowing parts from nothing. But even if you concede that you have all the parts necessary to build one of these machines, that's only part of the problem. Maybe even more complex, I think more complex, is the assembly instructions. That is never addressed by opponents of the irreducible complexity argument.
Studies of the bacterial motor have indeed revealed an even deeper level of complexity, for its construction not only requires specific parts, but also a precise sequence of assembly.
You've got to make things at the right time. You've got to make the right number of components. You've got to assemble them in a sequential manner. You've got to be able to tell if you've assembled it properly so that you don't waste energy building a structure that's not going to be functional.
Building a molecular machine has been compared to the construction of a house, where workers follow a detailed blueprint and plan for assembly. The foundation of a house is poured before the walls are erected. Plumbing and electrical fixtures are installed prior to enclosing the walls of the structure. Windows must be hung before siding is applied, and shingles are attached only after plywood sheets are nailed to the rafters. So it is with the construction of a flagellar motor.
You build this structure from the inside out. You are counting the number of components in a ring structure or the stator, and once that's assembled, there's feedback that says, okay, no more of that component now. A rod is added, a ring is added, another rod is added, U-joints added. Once the U-joints at a certain size and a certain degree of bend, about a quarter turn, that's shut off, and then you start adding components for the propeller. These are all made in a precise sequence, just like you would build a building.
To build a motor correctly requires a complex system of machines that coordinate the timing of the assembly instructions. But how could natural selection construct such a system?
The co-option argument doesn't explain this. You see, in order to construct that flagellar mechanism, or tens of thousands of other such mechanisms in the cell, you require other machines to regulate the assembly of these structures. And those machines themselves require machines for their assembly.
If even one of these pieces is missing or put in the wrong place, your motor isn't going to work. So this apparatus to assemble the flagellar motor is itself irreducibly complex. In fact, what we have here is irreducible complexity all the way down.
We know a lot about the bacterial flagellum. We still have a lot to learn, but we know a lot about it. And there's no explanation for how this complex molecular machine was ever produced by a Darwinian mechanism.
We have looked at the complexity of the genes that make up our body. not just the total DNA, but right down to the very pages of the book of the DNA language, couldn't have made themselves. They just suddenly appear, whole new pages, whole new genes in what should be closely related creatures, and vastly different genes in vastly different creatures, gulfs of gaps in information in the pages of life. We've looked at the impossibility that even the correct number or the correct type of amino acids could form by random chance. We've shown the statistical absolute impossibility that those amino acids could link up to form useful, proper protein sequences. We've looked at the unbelievable complexity of DNA and how DNA is not just even a simple language like sentences written on a page, but orders of magnitude and level upon level more complex than mankind ever even imagined when they first discovered that DNA carried the inherited characteristics of life. And lastly, we've looked at irreducible complexity, how unless all the parts are there and they're all placed into sequence and into position in exactly the right order in the right time, nothing will work. Level upon level upon level of evidence that leaves mankind without excuse for knowing that we have a creator. Now, there are many people who say, you know, creation's just a side issue. You know, as long as you believe in salvation and the Lord is your Savior, it doesn't really matter whether people believe in evolution and that's what they're taught. Well, how incredibly selfish. We have a whole generation that is literally and systematically being driven away from the trust and belief in God and the Bible because of what they're being taught in school and about evolution. How sad that Christians wouldn't want to know more and be willing to share the reality of who God is and the credibility of His Word in order to lead them directly to trusting Him as not only their Savior, but also their Creator. So, what is the excuse? How can very intelligent biochemists and biologists continue to believe that life just made itself based on scientific observation? Well, the excuse they hang on to, and that's all it really is, it's a misunderstanding of what is happening in nature, is the idea of natural selection. That if anything forms that has some sort of a biological advantage, gives the creature a useful function, then that will give the creature an advantage. He will make more copies of himself, and he will go on to take over some population or niche and evolve upward. He becomes more complex and more useful, and this will happen over and over again over millions and even close to 1 billion years of Earth history as chemicals came alive, turned into bacteria, bacteria eventually turned into people. Natural selection has caused it all to happen. Well, it's all fantasy, because natural selection could never cause information to increase, because the driving pool of information that natural selection has to choose from, and by the way, it doesn't choose anything. You see, the pool of information natural selection has to choose from are mutations, which are random changes to the genetic code. Like taking a book and randomly rearranging letters, taking a book and randomly rearranging words, taking a book and taking sentences from one place and sticking them in a different place. And supposedly that will increase the usefulness and information content of a given book or animal. Well, let's think about this. A bacteria has in the order, and this is very ballpark range, of about 1 million bits of information on its DNA code. The human DNA code has about 3 billion bits of information. It is literally 1,000 times more information
in a human being than a bacteria. Well, that makes sense. I mean, we have bones, we have skin, we have hair, we have a circulatory system, lungs, heart, organs, liver, stomach, all these things that aren't in a bacteria. Where did that information come from? Evolutionists are forced to believe, because they have no better answer, mutations to a bacteria, if they were useful, filtered by this thing called natural selection, caused the bacteria to form new features, more information to be added. Well, we've now studied that. We know what mutations do. And by the way, I don't know of any mother who says, I would like to relocate our house next to a toxic waste dump so I'll have a mutation for my baby. Or, I know, let's get lots of x-rays or live right next to an unshielded nuclear reactor so that my baby will be born with mutations. Because we know the vast majority of them are negative. Mutations don't make things better, in spite of fanciful cartoon books and movies. You see, as we've studied mutations, we find out that well over 99.9% of them are insignificant. They don't seem to have a positive effect, they don't seem to have a negative effect. Now in reality, they are slowly degenerating the information of the DNA code. Just like if you change one letter in a book with 1,000 pages, you could still read the book. It wouldn't seem to have a major effect, but the information's a little bit distorted. Same thing with a mutation. Now, probably of the number of mutations that might happen to an animal, and there's lots of mechanisms built into the cell to eliminate and prevent them, but some of them will be permanent changes. Maybe 9 out of 1,000 have a very negative effect and cause rapid deterioration of that creature. Now, what you'll see in textbooks are positive mutations, changes which have a useful function, and these can happen. A fish may be born where some of those fish thrive better in warmer water, so they tend to gravitate toward a warmer water area of a stream, and they'll form a whole new population in that area. A cave salamander may crawl into a cave and go blind from a mutation. Well, it now doesn't have to use its brain to function and process visual information, freeing up more of its brain to survive in the cave. That's called a positive mutation. But a blind man in a theater where the lights go out and someone yells, "Fire!" actually functions better in that dark atmosphere of that theater than people who aren't used to blindness. But no one would say blindness made that blind man a more advanced, better functioning human being. You see, it's just in a specific location it may have a useful function. Maybe once in 1,000 this might happen. So let's think about this. We are trying to build a mountain of information that isn't contained in a simple creature like a bacteria. Like, imagine Devil's Tower behind me. It's a huge 900-foot tall tower of information. That might represent the DNA information content of a human being. A little tiny bump at the bottom of Devil's Tower might represent the information content of a bacteria. How do we change that little bump into that tall tower? By random changes, mutations. Well, we're trying to climb up the tower. We're trying to build up the tower and go further and further and higher up. Mutations, as I said, are mostly neutral. They don't seem to have an effect. That would be like trying to climb this tower and we take 990 steps around the tower. We don't go up, we don't go down. We're right down there at the bottom level with the amoeba, the little bump. 990 steps around. But 9 out of those 1,000 mutations are very negative. So we take 9 steps downward, and then we take 1 step up, the positive mutation. 990 around, 9 down, 1 up. 990 around, 9 down, 1 up. Think about it. Are you ever going to get to the top of the tower? Are you ever going to build that mountain of useful information? The answer is so obvious. You see, God wants the truth to be known. There is no possibility that mutations and natural selection could increase the useful functioning information content of a low creature like a bacteria in order to turn it into a creature like a human being. It couldn't happen. And we know that because we know how these mutations and natural selection work. So the truth is on the side of the Bible. When Jesus said, "Have you not read, at the beginning I made them male and female." Human beings were made at the beginning of creation. There's never been these millions and billions of years, and evolution scientifically cannot and does not explain where it came from. But God's Word does.